Good day, students. The first lesson in Module 7 is about the structure of nucleic acids. So here we have the term nucleosides and nucleotides. When I say nucleoside, it consists of a base, a nitrogenous base, and a 5-carbon sugar, which could either be a ribose or a deoxyribose. A nucleotide, on the other hand, is a nucleoside which is linked to a phosphate group. So in nucleotide, we have a base, a sugar, and a phosphate group. So what is the nitrogenous base present in nucleosides and nucleotides? These are heterocyclic aromatic amines. When we say heterocyclic, we have more than one, or we have two or more different elements in the ring in the aromatic system. These are amines because we have carbons bonded to nitrogen. So in the nitrogenous bases in nucleosides and nucleotides, our parent compounds are pyrimidine and purine. So these are the two systems or ring systems or nitrogenous bases, parent compounds for our nitrogenous bases, I mean. So these are the 5 nitrogenous bases which we can find in our system now. Like for instance, in DNA, we have thymine. For the pyrimidines, we have thymine and cytosine. Whereas in RNA, we have still cytosine, and the other one, instead of thymine, we have uracil for RNA. For the purine bases, these are both found in DNA and RNA. The 5-carbon sugar in nucleosides and nucleotides could be D-ribose or 2-deoxydiribose. So for RNA, our sugar there is a D-ribose sugar. This is our D-ribose. This is the Fischer projection, and this one is our Haworth projection. For DNA, our 5-carbon sugar is 2-deoxydiribose. It's called 2-deoxy. This is our carbon number 2. As you can see here in 2-deoxy, the OH group is being replaced with hydrogen. That is why it's called 2-deoxy. So this 2-deoxydiribose, and this is the Fischer projection. This one is our Haworth projection for 2-deoxydiribose. Again, the hydrogen, the hydroxyl group in carbon number 2 is being replaced with hydrogen. Okay, so that's 2-deoxydiribose sugar. Again, nucleoside consists of a base and a 5-carbon sugar. So for purines, carbon 1 here is linked to the N9. This is the N9 for purines. In the process, in forming the bond we call here is glycosidic bond because we have our sugar molecule. So in the process, water is being released. Now, if we have a purine base, like for instance, we have adenine. When we bond or when we form a nucleoside using adenine base, the nucleoside formed will have osine as the suffix for the purines. If it's adenine, we have adenosine. And if we have guanine, the nucleoside form has guanosine name. For the pyrimidines, so this is our uracil pyrimidine base. When we create a nucleoside using a uracil base, the nucleoside formed will have an idine suffix. So the uracil, the acyl will be changed to idine, so that becomes uridine. The thymine will become thymidine, and cytosine becomes cytidine. Okay, so those are our nucleosides. Nucleotide again is a nucleoside which is linked to a phosphate group. So here, phosphoric acid is esterified to sugar OH group of nucleoside. That only means the phosphate is bonded or added to the CH2OH of our sugar molecule. Okay, so if this is our nucleoside, since we have an adenine base, the nucleoside name is adenosine. Now when we add one phosphate group, the name becomes adenosine 5'. This is 5' because the phosphate group is being added or bonded to carbon number 5 of the sugar molecule, we have monophosphate because we only have one phosphate group. So that's AMP, or adenosine 5' monophosphate. Now if we add two phosphate groups, the name now becomes ADP because instead of mono, we use the prefix di for the 2. And if we have three phosphate groups, we now have ATP, or adenosine 5' trimonophosphate. Here we have a summary of the bases, different bases in DNA and RNA, the names of the nucleosides and the nucleotides form. Here we have another summary, but we have now the structures for each of the nucleotides and nucleosides, deoxyribonucleotides and ribonucleotides. The functions of these nucleotides, so they have different functions. ATP, we all know that this is the energy currency of the cell. So we need this in our metabolism process, metabolic processes. GTP, on the other hand, is used for protein synthesis as energy source. CTP is for the lipid synthesis, whereas UTP is used for carbohydrate synthesis, polysaccharides. So what are nucleic acids? Nucleic acids are actually chain of nucleotides, or these are polynucleotides. No? So an example of nucleic acid is this one here. No? Here we have one nucleotide, we have another nucleotide. So when we connect these nucleotides, we have a chain of nucleotides, we form nucleic acids. Now if our nucleotides are ribonucleotides, the nucleic acid form is called ribonucleic acids, or RNA. And if our chain of nucleotides are deoxyribonucleotides, then we form deoxyribonucleic acids, or DNA. So nucleic acids are polymers of nucleotides. Again, these are linked 3' to 5' by phosphodiester bridges. These numbers here, 3' and 5', are the carbon number in our sugar molecule. So you can see the phosphodiester bridges, or the phosphate groups here, which are linked 3' to 5'. Okay, so these are linked 3' to 5'. Here we have shorthand notations for polynucleotide structures. So the furanoses, these are our sugar, are symbolized by vertical lines. We have our vertical lines here. Okay, these are our sugar molecules, or furanoses. The phosphate groups are symbolized by P. So these P here are phosphate groups, and the connecting lines between nucleotides are drawn diagonally. So these are what are phosphodiesters, no? These diagonal lines here. Again, the 3' and 5' are the carbon numbers in our sugar groups. So the primary structure of nucleic acid is just a sequence of the nucleotides. No? So this is divided in two parts. We have the backbone of the molecule. This P here stands for phosphate, S stands for sugar. So these are actually the backbone of our primary structure of nucleic acids, the phosphate and sugar groups. The bases, on the other hand, are considered side chain groups. Okay, and these bases here give the identity of that chain, of that polymer. No? So we have different side chain groups, or sequence of bases. Okay. Now, a simple notation of base sequence is merely to list the order of bases. So we use capital letters to denote the bases. A stands for adenine, G for guanine, C for cytosine, U for uracil, and T for thymine. Okay, so we only list the order, no, of the using capital letters for these bases. So the convention is to read the polynucleotide from the 5' end of the polymer to the 3' end. Okay, so if this is, for example, the 5' end, and this one is our 3' end. So when we write the sequence, we just write G, 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 and we have T, G, A, and so on and so forth. So there are two major classes of nucleic acids. We have the DNA and the RNA. DNA is found in nucleus, which contains the genetic information. RNA, on the other hand, can be found throughout the cell and has different functions. No? And with these functions, they can be categorized into 6. We have the messenger RNA, the ribosomal RNA, the transfer tRNA, small nuclear RNA, microRNA, and the small interfering RNA. So we start with DNA. The DNA consists of 2 polynucleotide strands which are wound together to form long, slender helical molecules. So we have double helix structure for DNA. So we have here, for example, we have our first strand and we have our second strand. As you can see, they are wound together to form a helical structure. Now this 3D model of DNA was established by James Watson and Francis Crick. Now this model is actually based on was formed based on two important pieces of information obtained by other workers. First, we have the Chargaff rule. This only means, simply means that the concentration of pyrimidine bases is equal to the concentration of the purines. Here, the concentration of A is equal to the concentration of T, whereas the concentration of G is equal to the concentration of C. This is actually in the table is the data of which leads to Chargaff's rules. As you can see here, we have 1:1 purine to pyrimidine ratio of concentration. The other information that Watson and Crick used for their model was the X-ray diffraction photograph obtained by Rosalind Franklin and Maurice Wilkins. So this is the structure or the X-ray diffraction pattern of DNA, and here the spots forming a cross in the center denote the helical structure. So they use this data for the 3D model of DNA. So DNA again has 2 strands wound together, and these strands run in opposite directions. So they are anti-parallel, and they are held together in the helical structure through interchain bonds. When I say opposite directions, the other one runs from 5' to 3', while the other one runs from 3' to 5'. Okay? So again, they are held together through interchain hydrogen bonds. These hydrogen bonds are what pair the bases of the nucleotides in one chain to the complementary bases on the other, and we call this base pairing. So here we have, for example, an unwound double helix. Again, as I've said, they are anti-parallel. So one strand we have 5' to 3', while the other strand we have our 3' to 5' here. No? So they are in opposite, anti-parallel. And again, we have our hydrogen bonding here. When hydrogen bonding occurs between hydrogen oxygen and hydrogen and nitrogen. Hydrogen bonding, here we have H between H and O and between N and H. No? So for adenine thymine base pairing, we only have 2 hydrogen bonds. We have 1, 2. Whereas for guanine cytosine pair, we have 3 hydrogen bonds. We have 1, 2, and 3. Okay, so we have 1, 2 for AT and 3 hydrogen bonds for GC pair. So the DNA in our body actually coils around basic proteins called histones. Okay, so if these are DNA double helix, they coil around basic proteins called histones. So DNA are acidic. So nucleosomes then are formed when our acidic DNA and basic histones combine by ionic forces. No? So we form nucleosomes. And then we form chromatin when the nucleosomes are wound in a solenoid fashion. And these chromatin fibers are organized into loops, so they become like this, and these loops are arranged into bands to provide the superstructure of chromosomes. So these are some differences in the structure of DNA and RNA. Again, in DNA, the pyrimidine bases are cytosine and thymine, whereas in RNA, instead of thymine, we use uracil. So in base pairing, here A pairs with T in DNA, whereas in RNA, A pairs with U. The sugar for DNA is deoxyribose, whereas in RNA we have ribose. For DNA, it's almost always double-stranded, which are helical, no? Whereas in RNA, they are typically single-stranded. So now we have RNA. Again, we have 6 types of RNA based on their functions. The first one is messenger RNA or mRNA. This RNA is produced in the process we call transcription, which we will discuss in the second lesson of Module 7. So the function of messenger RNA is that it carries the genetic information from DNA. So from DNA, it carries the genetic information to the ribosome and acts as template for protein synthesis. So we have here now our RNA. No? Again, this is produced in the process we call transcription. RNA consists of a chain of nucleotides, just like DNA. It has chain of nucleotides whose sequence is complementary to that one of the strands of the DNA. So for example, this is the strand of the DNA. The bases here in the RNA strand is complementary to that of the DNA. The second type of RNA is called transfer tRNA or tRNA. So this function as what? A transporter, no? It transports amino acids to the site of protein synthesis in ribosomes. So this happens in a process we call translation, and which we will discuss in the second lesson of this module. So here we have RNA, we have the amino acid here. So it functions to bring the amino acids in ribosomes in synthesizing proteins. So this is a 2D structure of tRNA, and this one is our 3D structure of tRNA. Now there is at least one different tRNA molecule for each of the 20 amino acids from which the body makes its proteins. We have different types of, different molecules of tRNA. The third RNA type is called ribosomal RNA or rRNA. So from the word ribosomal, this RNA is complexed with proteins in ribosome. So they make up, no? Together with protein, these RNA make up the structure of ribosomes. So our ribosomes are 35% protein and 65% rRNA. And we know that ribosomes is the site of protein synthesis. So this is our structure of ribosomal RNA. So that structure occurs because of the hydrogen bond interactions, no, of the bases in the structure. The next type of RNA is called small nuclear RNA or sRNA. So this is found in nucleus of eukaryotic cells. So what does it do? This is actually complex with proteins to form SNRPs or small nuclear ribonucleoprotein particles. So this helped with the processing of the initial mRNA transcribed from DNA. Remember, in the process of transcription, we form mRNA. So these SNRPs help to process that mRNA into a mature form that is ready for export out of the nucleus in a process called splicing. So the mRNA formed from DNA, or in the process called transcription, will be processed. No? So there will be splicing, and that occurs with the help of SNRPs, which contains our sRNA or small nuclear RNA. The next type of RNA is called microRNA or miRNA. This is small with 20 to 22 nucleotides present, and this is important in the organism's development. No? It plays vital role in cancer. So what it does it inhibits translation of mRNA into proteins. It could help promote the degradation of mRNA, for example, in cancer cells. No? And it stimulates protein production in cells when the cell cycle has been arrested. So it can be used in stress responses, or to prevent viral infections, or to not prevent, or to stop viral infections. The last type of RNA is called small interfering RNA, or siRNA. So this is have enormous control over gene expression. It can be used to eliminate expression of undesirable gene. Example, the gene that causes uncontrolled cell growth, as in cancer cells, for example, or one that came from a virus. So this leads to the degradation of specific mRNA molecules. So here we have the summary of the different kinds of RNA and their functions. Again, tRNA is used to transport amino acids to site of protein synthesis, or the ribosomes, in the process called translation. Ribosomal RNA together with proteins is what form our ribosomes, which is the site of protein synthesis. Messenger RNA is formed again from DNA in a process called transcription, which then directs the amino acid sequence of proteins. The small nuclear RNA is complex with proteins, which are used to form SNRPs, which are used to process the initial mRNA in a process called splicing. MicroRNA and small interfering RNA both affect gene expression. MicroRNA is important in growth and development, and this small interfering RNA is used by scientists to knock out a gene being studied.